Hi, teacher. I am Alex Valdivieso, and today, me and my partner, we are going to talk about the lab practice number 5, the surface swab. Well, in the realm of food safety, ensuring the microbiological integrity of food contact surfaces is paramount. This is where surfaces swabbing, a fundamental technique in food microbiology, takes a center stage. By meticulously collecting samples from these surfaces and subjecting them to rigorous laboratory analysis, we can unveil the hidden world of microbes that may pose a threat to food safety. Surface swabbing is an essential technique in microbiological monitoring of food processing environments. Through this technique, it is possible to collect samples of microorganisms present on various surfaces, such as work tables, utensils, personnel, and process equipment. These microorganisms can include bacteria, yeasts, molds, and in some cases, viruses, depending on the culture medium available, which can be indicators of processes. Objectives. The general objective was applying swabbing techniques to detect microorganisms on surfaces in food processing plants, emphasizing the importance of hygiene and safety control. The specific objectives perform surface swabs effectively using aseptic techniques, cultivate and identify microorganisms present on the surfaces of processing plants, understand that relationship between surface hygiene and food safety, critically analyze the results and their impact on the food industry. Materials and methods. For this practice, we were using sterile swabs sterilized in the autoclave, preferably with a cotton tip and non-wooden handle. Culture media plate count agar, PCA, petri dishes for the cultivation of microorganisms, 4 per group. Sterile saline solution or Pepton water to moisten the swabs before sampling, dilute the samples and prepare according to laboratory availability. Disposable gloves, apron cap, mask and closed shoes. Incubator, felt pans, disinfectant and cleaning material. Burners or laminar flow chamber methodology. Preparation. Uh, First, organization of the group work in their laboratory groups, assign specific roles for each member, such as sample collector, taking picture and data, and cultivation managers for each crop. Make sure the swabs are sterile and ready to use. Sampling. Surface selection. Choose at least 2 different types of surfaces in the processing plant to sample, such as work, tables, door handles, and equipment. 2. Softening. Lightly moisten the tip of the swab in the sterile saline solution or Pepton water. Swab an area of approximately 10 centimeter for 10 centimeter, with the swab inclined at an angle of 30 degrees. Rub the surface delimited by the blade 4 times, each one in the opposite direction of the previous one. Ensure the swab on the entire surface. 3. Labeling and recording. Mark each petri dish with the date, sample location, and group. Record all surfaces sampled and conditions observed during sampling. Finally, sowing and cultivation. First, sowings 1 milliliter of the isopus rinsing material. In case you have cavitation during repeating, use a smaller now balloon. You can dilute it to 10 elevated to less 1 or so directly by the extension method. 2. Incubation. Place the plates inverted in the incubator. Incubate at 35 Celsius degrees for 48 hours. 3. Analysis of results. For regular surfaces, the number of colonies obtained CFU will be multiplied by the dilution factor and by the volume of diluent solution used in sampling, 10 milliliters, and will be divided by the area of the swab surface or sampled, 10 square centimeters. Results.
Analysis of results. The first part is the the structure of the hand area. There are several results of cases of people with smaller hands that contain less data, where they are less contagious than those with large that, that have more contact surface. And the next part is the culture media, is the plate count agar PCA, also called standard method agar is SMA, is a microbiological growth medium commonly used to assess or to monitor total or variable bacterial growth of a place. PCA is not a several medium. The next part is bacterial count. We found two fungal colonies right on the plate contact agar, PCA agar. So got excellent results from the patients that there is not much contained and there is cleanliness control. And the colonies found. We found about 3 colonies in this plain culture, the number 2 of the hands, right, of the right hand, which saw that a certain part of the population that washed the surface of their hands quite well. Okay, and conclusions. The involvement of a laboratory in performing hands swabs in restaurants and food restaurants is essential to ensure public surface and hygienic. These swabs are a crucial preventive measure to detect the presence of pathogens and maintain the health of the population. By tasting sample from hands and surfaces in restaurants, bacterial, viruses, or other microbiologics that could cause foodborne illness or other infections can be detected. Okay, annexes. What is the main purpose of surface swabing in food processing plants? Okay, the main objective of surface swabing in food processing plants is to detect and control the presence of microbiological and physical contaminants. This test identifies areas where bacteria, viruses, and other pathogens accumulate, allowing corrective measures to be implemented to improve cleaning and situation starts. This ensures the production of food, completing with synergistic relations and maintaining consumer confidence in the equal of food products. Okay, the second question is, describe the correct processes for taking a swab sample. Why is important to follow this method? To take a swab sample, sterile equipment and disposable gloves must be used. The swab is maintained with a sustainable transport medium and rubber film over the surface to be sampled. The swab is then placed in a sterile container and sent to the laboratory for analysis. It is crucial to follow this method to avoid cross-contamination and to guarantee the accuracy of results, ensure a reliable assessment of the hygiene and safety. The third question is, list 3 types of surface that will be important to sample in food processing plants and explain why. Okay, the crucial surface to sample in a food processing plant are detected products contact areas, such as table and equipment to avoid contamination. In addition, the environment including floors and walls should be sampled to prevent the spread of pathogens. It is also essential to sample heating and packing surface to ensure that the final products remain free of contaminants and safe for composition. Okay, the next question is, what type of cultural media is used in this practice and food? And for what type of microorganisms it is the best suited? In this practice, selective and differential culture medium, such as nutrient agar or support agar, which promote the growth of specific microorganisms and allow their identification, are used. These areas are suitable for a wide range of microorganisms including bacteria and fungi knowledge associated with food contamination, such as Salmonella, Escherichia coli, and Spirlicus species. They have detected the presence of equal of microorganisms in samples. And finally, explain the sample incubator process. Why are the invented Petri dishes placed in the incubator? The simple incubator process involves placing Petri dishes with culture media and samples in an incubator at a controlled optical temperature for the growth of microorganisms. Petri dishes are placed inverted to avoid condensation of water of the surface of the media, which could interfere with bacterial growth. This method also prevents eternal contaminations and ensure accurate results during the incubator period. This is all for this presentation. Thank you so much.